Hi everyone, welcome to this session. I'm so sorry that it took a bit longer solving some technical issues, and now we're back. And I'm very glad to welcome Dr. Evelyn Tang. She is a group leader at the Max Planck Institute for Dynamics and Self-Organization in the Living Matter Physics department. She completed her doctoral work in condensed matter theory at MIT and then worked as a postdoctoral fellow in the field of complex systems with Dr. Danielle Bassett at the University of Pennsylvania. And today in her talk, she's going to talk about the geometry and topology of neural systems. Just a few technical things before I give it off to her. You can ask questions using the Ask a Feature question on the bottom. Please indicate whether you want to be invited on the screen or not. Okay, then I give it off to Evelyn. Thank you.
Great. So you can hear me all right? Thank you so much for the invitation for this really interesting meeting. It looks like such a wonderful collection of different topics and speakers and, great organization. So thank you so much. I'm very happy to be a part of this. So I suppose I'm a physicist, so I'm coming into this from a more physics perspective, thinking about how can we use some physics tools to analyze and understand the very complex system of the brain. So this is just an overview of different things that I think about. I've worked a lot on brain networks and control, effective learning. You can see my cursor, right? And my PhD was on topology. And today I'm going to be talking mostly about learning, and if time permits, about some new work that we have on information in fluid flows, for example, in brain ventricles. So first, I'm going to start to introduce learning, and of course, this conference is organized by students. Learning is a huge thing that we do every day, especially now in let's say coronavirus. We're all having to learn how to deal and adapt with the virus. So learning is something we do every day, and not only is it central to our lives, but as our technology is improving so rapidly, and we now have robots and artificial intelligence systems, can we also understand better what the learning process is so that we can use that to teach robots what to do, for instance. And some might already say, well, we already have, a paradigm for learning, which is machine learning. It works. It definitely works very well. Self-driving cars seem to be right around the corner. But at the same time, given that, many of us will be partial to the brain, let's look at what are some of the costs of machine learning, which is that firstly, there is a big cost to the environment. Of course, it uses a lot of energy, as you can just see in this benchmark paper that came out last year, just in the cost of training a new model. For example, here, this is one of the new models, and of course, they've been getting more complicated over the years. It uses a lot of energy just to train the model compared to, let's say, an average human life, an average American human life, lifetime of a car. And this is just to train the model. So let's say the first few days to get the model started up, and then there will be additional costs to run the model, to fine-tune the model, to retrain the model for different purposes. And not to mention that these models have a huge number of parameters. And so that already is something that just from a theoretical point of view doesn't even make sense, because oftentimes we don't even have that much training data to offer to these neural networks. And so then are we overfitting? What actually are these parameters doing? It becomes very difficult for us to look into these parameters to understand any structure that these networks are actually coming up with. So in some sense, can we actually understand what we are learning from these networks? And let's contrast that with the human brain, which is comparatively quick and efficient. I'm sure this is evident to all of us. I just have this picture in here because it makes me laugh sometimes when I see, for example, a baby, let's say a 1-year-old child who can barely walk or talk, and yet they are interacting with a cell phone in a really fascinated way. And it's presumably because they see their parents really interacting a lot with the cell phone, and so they're also mimicking this behavior. And so really we see that kids will pick up whatever they see, whether or not we even guide them towards doing it. People learn things very naturally and quickly and efficiently without even now having to sometimes prompt them. And so what are underlying principles of learning? If we can understand better biological learning, perhaps this can start to unlock some of the mysteries instead of us having to throw all this very brute force computational resources at the problem. However, to understand biological learning, especially humans, of course, we know it's difficult. I'm a theorist, but I know our work relies very much on many experimentalists out there who take data, and this data is often noisy, and it's very hard to do control experiments in human beings, of course. And then because fMRI data tends to be expensive, the sample sizes are small, and most of all, we don't even have a theory to compare what our data comes to. So then, from my point of view as a physicist, I'm going to try to look for coarse features or cause autoparameters in neural data to help us to distinguish between different cognitive states and between different effective learning states. And so what I'm going to be looking at are a coarse-grained feature of a multidimensional data set. And multidimensional data set, because we're going to be looking across the whole brain, since learning engages so many complex dynamics, it's sensory, attentional, memory, which are different time scales. As we learn something, we see and listen, and then I try to synthesize it using my attention. And then, of course, there's the memory that comes over days and over weeks and months. And so as I've told you, given the noisy data, we won't study sort of a very specific dynamic and a specific voxel. But what I'm trying to look for here is maybe a coarse feature. And in this case, the idea here is we're going to look for the right dimension of the data. And this is a simple idea that we're all used to in just thinking about how to extract the right structure or the right curve from a bunch of data. And you can see that if you use too few parameters, you run the risk of underfitting. If you use too many parameters, then you might capture all the data, but then you often capture probably a lot of noise, and it's difficult to generalize when you have new data. And there should be sort of the right, Goldilocks range of the right amounts of parameters or dimensions that will allow you to capture the trends without sort of capturing spurious information. So our hypothesis is that there exists a suitable dimension, this is a coarse feature we'll be looking at, that allows us to capture the computational complexity of the task that we are being learned. And this task is definitely going to be set by what we are learning and not the parameters of the neural network or of the imaging of our hundreds of thousands of voxels that we have access to. But it is really set by the task. And so just to give an example of what I mean by this, in this classic experiment that's almost 20 years old now at MIT, where they trained macaques to categorize between pictures of cats and dogs. And once the animals were trained, they could see that the neural patterns actually, in response to the cats and the dogs, could be separated by a linear classifier like so. So you can see that in this case, that there was an emergent dimension along which the data now could be separated. Before, maybe the data was all, just some hairy ball of data, as we often get. But then after training, the neural responses then could be separated along this axis, so along this new emergent dimension. And this dimension is then representative of the task that the animal has to perform. Of course, in real life, we have not just one single dimension, but many, many dimensions. And so then the idea will be to capture that right dimension that's appropriate to the task. And at the same time, we know that when we measure data, of course, we get a much, much higher measurement space, because let's say we have many voxels or many probes. And so here I just want to highlight the fact that as we know, the data often lies in a much smaller space. That if you look at the huge measurement space that we lie in, the actual variance that we have is usually just along a few axes, or it's much smaller than the actual possible huge measurement space of the data. So just to drive home this point, this picture, I think, sort of captures it, that here we can have 3 neurons, and the measurement of the third neuron may be just a linear combination of the first 2. Whereas in this case, the third neuron can give us information that's actually different from the other 2. So this picture has a higher dimension, higher effective dimension, and this one has a lower effective dimension. And so what we really want to do is to look at neural data and see if we can probe the effective dimension of the neural data and see what that has to do with the task that the participants are being asked to perform. So please feel free to interrupt me at any moment for questions. And then we want to link and see if this dimension is connected to successful learning. So here we're going to be looking at a high-dimensional data set, and how can we estimate the dimension of that data? So I'm going to be using this combinatorial approach, and it goes as follows. If we have a certain number of categories of data, so in one example that I showed you, let's say cats and dogs could be different categories of the data, and for each category you get many different data points as responses to that particular cats, for instance. And so what we're going to do is to assign binary labels to the data. So some of the categories can be called blue, some of the categories can be called red. And then we'll look at how easy it is to separate the blue from the red. So you can see that when the data lie in a one-dimensional arrangement, that there will be some assignments in which we can separate the blue from the red, and there will be some other assignments in which it is difficult to separate them. When the data are lying in a higher dimensional space, it becomes then there will be more binary assignments on average in which it is possible to separate the data. And then so So just the total number. So then we're going to go through all the different possible assignments of binary assignments of the data, and we look at on average how easy is it to separate these binary assignments, and then just the average success of the linear separability across all the different assignments will then be an estimation of the dimension that the data lies in, that the data forms. I hope this makes sense. If not, you can look at this paper in which this method first came out. And this method is going to be better at PCA, especially in this case where we don't a priori know what the underlying metric of the solution is. For example, we don't know whether or not this measurement probe should be as high as it is, or whether it's actually not really relevant. So in this case, since we don't have an underlying correct metric in which to embed the space, then this is an approach that can then capture the right dimension of the data. So we're going to apply this to an experiment with humans in which they had to distinguish between different stimuli. And these were computer-generated stimuli that were generated to have the same statistical properties so that there wouldn't be any a priori association of the shapes with the value that they were assigned by the experimenters. And so these shapes had all dollar values assigned. There was a Gaussian with a mean at that particular dollar value. And the participants had to learn to associate these dollar shapes. And they were trained like so. So they were, for each trial of the experiment, and they had to do this 1,600 times over 4 days. And for each trial, they were shown 2 shapes at a time. They had to make a choice. So in this case, let's say the participant chose the right shape. They were then given feedback on that choice, and then blank screen, and then the next shape, or the next pair. And this went on for 4 days. And by the end of the second day, all the participants, we had 20 participants in the study, had learned the shapes well. So, and at the same time, while they were learning and they were being tested, where each session had 140 pairs shown, the fMRI data was also being measured, and this was being measured across the whole brain, as I said. And because we're interested in looking at the whole brain here, so here then we use the coarse-grained approach of using a large whole brain parcellation. In this case, we use a Lausanne parcellation of 83 regions to look at the aggregate time signal response in each of these 83 regions. We can then from the time series, we can then extract the responses to each of the shapes as they're presented by deconvolving the data using the typical hemodynamic response function to obtain the static value of the activation response to each shape for all 83 regions. We can then put that in our 83 region space so that each point, each response, each neural response to a shape that's being shown forms a point in this 83 region measurement space to form a 140 point data cloud. And now we can just test or measure the dimension of this data cloud. And we're going to measure the dimension of this data cloud by using this method that I had explained before about assigning binary labels and going through all the possible assignments and looking at, on average, how easy is it to separate these different binary assignments from each other. I know this is a little bit fast, but this talk is being recorded, so you can always ask me if there's any questions. So, but in the meantime, and please let me know if there's any questions. But otherwise, I'm going to keep going. And then now we can look at some of our results. So what happens when we look at the dimension of this data cloud for our individual participants? And as I had told you, by the end of the second day, they had all been doing pretty well. This is the accuracy. But actually, what was different among the participants was at the end of the first day, there was actually quite a large variance. Some of them had learned this, and some of them had not. So we use their performance at the end of the first day as a proxy for how fast they learn. And this is going to be now the x-axis of our plot, and then the dimension of the neural responses of each individual, as I had explained before about how we got it, is then the y-axis, and so that we can see whether or not, let's say, a fast responder has a high dimension, and we see that indeed they do. And at the same time, we also did a null model because, again, here we don't have an underlying theory, so we have to use an empirically or numerically generated null model. And we do that by taking the same data so that it will have the same properties, same sampling, same whatever properties. And now we are shuffling the shapes, the shape labels between the different data points. So that now we have the same data cloud, but now all the task labels, all the shape labels are now shuffled, so that now there should be no task information left in this data cloud. But then now we can repeat our analysis and to see what happens, because this then can then be a null model from which we can compare whether or not our result is significant. And what we see is that when we do shuffle these, this data cloud, in this case we had 1,000 bootstrap samples, we see that they form this more or less Gaussian, and this is our true data that we had on the left, and we see that the true data definitely lies outside. So this result is significant. So this is what we did just to see if our result was significant. However, what was interesting was that when we then looked at this null model some more, we actually found some interesting structure. So this is the result that I had told you before that we had from the true data, in which we found that there was a positive correlation between the accuracy, between the performance of our participants and the neural dimension. So essentially, our participants, the ones who learned the fastest, had a larger dimension of the task responses data. And we thought that the null model would actually, it shouldn't contain any information, so it should be around 0. But at the same time, instead what we saw was that the null model actually showed something different. It showed the opposite. It showed a negative correlation. So what this means is that the null data, the data that doesn't have any information of the fast learners, is actually smaller than the null data for the slow learners. And this was a really big surprise. And we really had to take this seriously, because actually as we increase in M, that's the number of shapes that we were including in our analysis, because it gets computationally pretty expensive. And that means that we're now averaging over a large number of combinatorials as we go to large M. So these are actually the most reliable results. And we saw instead that the null model had this negative correlation. So in fact, now we have two pieces of information about what our fast learners do, that the fast learners have both a higher dimension of response to the task and a lower dimension of just, neural data in general. And so these two pieces of information are consistent with these two ideas of dimension that I had explained before, that you want sort of enough dimension or enough parameters to capture the computational complexity of the task. So you want sort of a large dimension for the task data, but you also don't want sort of like too large data of just the neural responses that you use to code for the task at hand. I mean, if you're just trying to code for 12 shapes, you don't necessarily need enough neural space to code for 20 shapes, or you could, but it would be very inefficient. And in fact, we saw that this null data, or this data without any task information, was actually larger for the ones who were learning more slowly. And so we actually realized that there could be an efficient, what we call an efficient representation, which here I'm sort of using the little cartoons before, just because it's very hard to visualize data in very high dimensions. So this is the sort of highest dimension we can do for visualization. Then we have an efficient learning, and we have inefficient learning on the right. And an analogy that I can give is an efficient learner is someone that, say, for example, this has the kind of organization that you would have if, let's say, you had all of your papers on your desk sorted in different piles according to the different categories. So then, if you have that organization scrambled, then you have all the papers sort of mixed up on your desk. But what would be really inefficient would be to have all that data scrambled up, but not just on your desk, but in your entire room. So not just do you not have any order according to the actual structure of your task, but now you're using a bigger space in which to have that disordered information. So it's even harder to then find something because you have so much space. So I hope this analogy helps, and these little cartoons, I know they're just cartoons, but overall, these two pieces of dimension help us to reflect that there can be different types of dimension that contribute towards an efficient representation, which means a high information coding, but a small amount of resources that are used for the coding of the task, which is then makes it easier than to find the information. And you can find more details in our paper that came out last year. And so all of this was a study across the whole brain. But then, of course, look at on a smaller time scale. I just want to also discuss this other paper that came out at the same time, around the same time, that was looking in neural networks, so not at human brain data, but they also analyzed the shape of these manifold representations in neural networks, and they also saw two different kinds of dimensionality that could change in different ways for training. So in some senses was analogous from a totally different method of analysis. And then, as I said, we can also then use this as a data-driven approach then to ask, well, we look at the whole brain, but which region in the brain contributes the most towards the result that we saw? So to understand this, we looked at a virtual regioning, where we removed one region of the brain at a time, and then we repeated the analysis to see which region changes the results the most. And what popped out was the left hippocampus, which has particular associations with the rapid learning of stimuli, and the right temporal pole, which codes for more abstract conceptual properties. So this was a nice way in which to see whether or not our work could be consistent with already known results in the neuroscience literature. And we can also see whether or not this result of increasing dimension according to the task, which we saw in the whole brain, was recapitulated on a smaller scale, let's say in particular regions, but now on the voxel level. So here we looked at 5 regions in each hemisphere, and we did indeed see that either the dimension remained constant across the task or it increased according to the accuracy, depending on the accuracy of the participants. And these were the regions that, for example, in which we did see that there was this also increased dimension for the fast learners as well, which are the anterior cingulate cortex, as well as V1 and posterior fluvium. And all of these regions have something to do with coding for reward learning, which is what we were trying to look at in this task. We didn't want a task that only picked out visual cues, but we also wanted to connect it to the sense of reward by assigning actual dollar values in this task. And in fact, the participants were compensated not just for the time, but also in addition, they were also compensated based on their performance in these tasks. So there was an incentive for them to also learn the values as quickly as possible. So I'm just going to summarize this particular section that we like so. So fast learners have a higher dimensional representation of the neural activity. This allows objects of different value to be more easily distinguished from each other, because then so the neural response then will be in a different space mentally. And they also have a lower embedding dimension, which means that overall they have a more efficient representation, which allows them to code for a lot of information, but use smaller amounts of resources in doing so. And overall, I would consider this as being a more geometrical approach to looking at learning and cognitive performance. And this is why I was saying that coming in as a physicist, the goal here is really to try and think about using different kinds of tools and analysis in order to capture some of the interesting aspects about cognition that may be present. So this is the end of the first, the main part of my talk that I wanted to go through. I know I may have been a little bit fast at times. I don't know if we should take questions for this part now, or I should maybe just go through a little bit for the second part, and then we can take questions all together, or what do you think is best?
We can go ahead right now and then take all the questions in the end.
Okay, great.
Yeah.
So this is where we are, and this is what I was talking about, and then I'm just going to be talking a little bit about some new work. It's quite different, but I think it actually connects pretty well with some of the other talks that have been going on. For example, I really liked Esther Stokely's talk about the role of the primary cilium in sensing, and I think more and more we are seeing now that there's a lot of molecular signaling that is happening that is leading to the development of axons and on brain tissue as well. So this is a different task. It's looking a little bit more at morphogenesis and development, but hopefully it can also spark some interesting discussion. And we actually started to look at this problem because there has been very nice work in the collaboration between my institute, the Max Planck Dynamical Systems, and our neighboring institute next door, MPI-BPC, which who are for kindly organizing this wonderful event, in which they were looking at, they have been looking in the brain ventricles of mice, and they've been slicing them open, and they find that in the ventricles of these mice, they see very complex flow patterns where, for example, this may change even depending on the physiological state of the mouse, where in one case you see some kind of whirl, in the other case, actually you see some sort of more straight flow. And we know that the flows that are being transported in these brain tissues, the cerebral spinal fluid contains guidance molecules that cue the migration and development of young neurons, for example, in this paper, but also we've definitely seen that in Esther Stokely's talk as well. And so our question was, well, can we try to understand these very complex flow patterns and understand what they can tell us about the information that can be contained or carried in these flows? And so the tools that we'll be using to quantify information in these systems is now to look at information that is being carried by, let's say, small fluctuating particles in a complex flow. And this is interesting also from the point of view theoretically, because a lot of times these particles are so small that there's a lot of fluctuation. So they can really, even if you have a flow pattern, they can move, they will diffuse, they can move aside very quickly. And so how can the system reliably obtain information despite the fact that they're using such small particles, whether it's neurotransmitters, proteins, genes, they're all very small, but yet they can allow for chemical signaling very efficiently and very predictably in organisms. And so here, the way to quantify information that we'll be using is that of Shannon entropy, which in these flows we can look at by looking at the mean of this log entropy for the system. And Shannon entropy is very intuitive in the sense that when the probability distribution is very sharply peaked, then this system entropy is low, and which means that we can then reliably locate where the particle is in space, because we know that it's very likely going to be at the peak. And when the particle is very spread out, very diffuse, then this entropy is high, and this means it's a lot of uncertainty on where the particle is. So these differences in entropy can then tell us about how much information we have about where the particle is. And in fact, if we look at just the diffusion of a single particle without any flow, we see that it's really simple. It just depends on the dimension and over time. And so here then we can then look at the results of entropy production in a flow field by using the continuity equation, and we look at what its contributions are. I'm just going to go quickly through the math, and then we can even look at the heat dissipation in the medium, so the entropy that's produced in the medium. Interestingly, we can see that for certain kinds of absorbing flows, there can actually be a kind of cooling effect that happens from these flows, even if the total entropy is of course always increasing, it's always positive. And then so then we can then look in sort of very complex flows and look in particular in little neighborhoods by using a Taylor's expansion, and we can solve for the probability distribution of that particle. And then we can also ask for what is the residence time. So essentially how long, or if you have a particle in a let's say a very complex flow, is it going to stay here for a long time? Is it going to get washed away very quickly? And what happens when it reaches, let's say, a complex region like this? Will it stay here, or will it move away very quickly? We wanted to know how long will the particle remain in that region, so that we know whether it has a chance to bind or unbind, let's say, to the tissue or the axons or whatever may be receiving that information from that particle. And so here we estimated using a formalism, a time scale that depends on the speed, and of course again on the divergence of the particle, whether the fluid is absorbing or whether it's secreting. And so then we can test these predictions by looking at, again, flows that now are absorbing, and this one is a secreting flow. And this is just the flow profile. And we can see that actually as we expect, that our predictions actually suggest that these absorbing flows then retain the particle, and these secreting flows delocalize the particle. And we can see this in the information that we calculate, that these absorbing tissues then have these much more information than these secreting tissues. And then we can also look in flows that have some kind of rotation, some kind of vorticity as well. We can see that it has a shorter residence time and a different change of information. I know I'm going a bit quickly here, but I just wanted to, show you some of these things. I know it's a little difficult to go through all the math now, and we can definitely discuss more over questions in a couple of minutes. And then we can also look at what happens if you just look at a pure vortex. And interestingly here, we actually see that this kind of flow has, the same kind of information profile as just pure diffusion, but now we get very nice oscillations because of this vortex. And so this is really a place in which we actually see that it could be a way of capturing particular transmitters or signaling molecules in the vicinity of that vortex, which just goes around. We can also then apply this to more complex flows. And, let's say, because this is a formalism that can be used for a flow field that in different of arbitrary kind, we can apply a formalism for that particular neighborhood. And we see that even for, let's say, here with two vortices, we find different results for the localization and for the information. And we can also find some effects of some temporary refrigeration as well, which may or may not be happening. So in some kind of sucking or absorbing fluids, maybe this could also be a cooling for the system, which may be something that we could test experimentally. So I'm just going to summarize this particular section to say that we are now here trying to quantify entropy production And I think this is a step towards really recognizing that there's a lot of stochasticity in the underlying components in our neural systems, and can we try to build a stochastic formalism that incorporates these things so that we can then try to understand better how the stochasticity still emerges to give some robust functioning. And here then we identify which are the most relevant flow properties, where it's strain instead of rotation, and when something matters. And we can apply these formalism to realistic or measured flows, and hence this could be relevant for signaling processes, such as the ones that I had mentioned in the introduction of this section. So I want to acknowledge the people that I did this work with. My postdoc mentor, then Professor Chad Giusti, was one of my favorite collaborators at Penn. We talked a lot all the time and came up with a lot of these ideas for kinds of analyses that we could do. And of course, my current group here at the Max Planck for Dynamical Systems, headed by Ramin, and of course my wonderful departments, my students, Laura and Lorenz. So I'm very happy now to take any questions if you have any.
Okay, great talk. We have some interesting questions, and I will start with Mathis's question. He asks, regarding the first part, if I understood you correctly, you said that the method that you are using is better at assessing dimensionality than PCA when there is a lot of noise. Sorry. Just out of curiosity, did you also try to assess dimensionality for the learning experiment with PCA? If so, were the results similar?
So we did not try and use PCA. We did use some other things. For example, this is a very computationally heavy approach. For example, we just use a kind of vanilla linear classifier, and we did compare there and there. Let me see if I have that slide. Yeah, here it is. So here actually we did look at just comparing it to the linear classifier across all labels, which we called assortivity, and we can see that actually in this graph we can see that dimension which we're using and assortivity kind of measures different things, because something could be, let's say, high in assortivity and yet low in dimension, and vice versa. And we actually saw that while it does capture some things, one metric explains not a huge amount of variance of the other. So there's not really a straight overlap. So I think that a dimension is capturing something different. And what I was saying about PCA is because let's see where the slide is, that we didn't want to use PCA because PCA, sort of assumes that for example, all of your measurements approaches, all the axes are orthogonal to each other, and also the numbers matter. For example, if you rescale one particular measurement axis, it will change the contribution of that very strongly. So really, the metric matters, and whereas in this approach, it matters. It doesn't matter as much because you're looking at distances between different groups of the data. So in some sense, even if you rescale an axis, it doesn't change the results so much. And also then what I was saying about how PCA assumes that you have orthogonality. I mean, this really assumes that, for example, all the different brain regions are independent of each other, which we know is not true. I mean, a lot of them are correlated. So for many reasons, we didn't think that PCA could really capture the kinds of dimension that you have. I mean, for anyone who's worked with neural data, often you see that neural data is a hairy ball. It's a very messy thing. It doesn't have sort of like nice dimensions on nice axes in which you can actually see structure. And so I think PCA works well when you can already see, when there is already some kind of structure, and it's going to pick out the structure, it's going to pick out the variance that you have. Whereas if you just have some hairy ball and really, it doesn't look like it has structure, then PCA isn't really going to help you. And so because of that, we didn't try to use PCA for that. We wanted to use something that really could discriminate a little bit better, even with just like a hairy ball of data.
I think there's a clarification question from Yanin. Okay, there's a clarification question from Yanin. She asks, could you repeat how the findings of part 1 and part 2 of your talk are connected?
So, I mean, they are not necessarily connected in the sense that I think, I saw Matthew's statement about liking the hairy wall metaphor. Yeah, I mean, sadly, a lot of times, you you look at the data and you're like, oh my goodness, what is this, right? That's usually my first reaction. So back to
For me, that's the most exciting part.
The most exciting part. Yeah, that's great. Yes, you're committed. Well, I think that's really, that's what I would say is, let's look at, that's, well, I mean, they're connected in the sense that that here I'm really looking at, let's say, adaptive processes, that they're connected in the sense of, here they're sort of on this side of the time axis, in which learning is an adaptive process. I see something, I want to understand it, I move towards it, and then once I understand it, usually the conditions change, or I want to learn something else. So I'm really constantly moving and adapting. And when I'm looking at information and fluid flows, really flows are moving, they're changing. And so here I'm also, and so therefore the particle, as you can see here from this little cartoon of the Gaussian, it's going to spread out. So this is also something that's changing with time, that's transient, and that's really going to change and adapt depending on how the fluid changes, on what the signaling needs are. So here, I suppose one connection between both of them is that we are looking at adaptation, and I'm trying to understand really how trying to build tools to understanding what are some of these dynamical processes in the brain, which is something that is really constantly changing all the time. And I would say that this is also, in some sense, maybe a little bit of a different approach from a physicist, because as a physicist, one thing that's nice about us is that I'm a theoretical physicist. So, in some sense, I can also use different approaches and different ways of thinking, even for the problem about cognition. So I think that's something that I feel very, very lucky. It's both good and bad sometimes. There's so much space, but because of that, I guess my work hasn't been necessarily, let's say, only about learning, or only about one specific topic. But in some sense, the goal is really to try and develop different tools. And ultimately, I don't really think we're there yet. But ultimately, it would be great if we could understand a bit better one day how things scale up from the microscopic scale to the macroscopic scale. And so that's also why in this slide, I also have this axis on space, because a lot of times when we think about cognition, we look at it from the more macroscopic scale. We look at it from what are broad patterns that are being learned, which is necessary because learning is something that uses so many different modalities. And also the imaging that we have is usually a voxel is probably, let's say, sub-millimeter, or I mean, it's usually already considered sort of mesoscale, or sort of is not microscopic. We really, it's very hard to ask, if you want to ask about human learning, what's happening at the neuronal scale. At least if you want to use non-invasive methods. And so then another, I think, somewhere in which neuroscience can move towards, and hopefully will move towards, is to connect more the microscopic, like neuronal firing, cellular signaling, chemical signaling, up to the macroscopic scales that we see. So we're not really there yet. We don't really know of the bridge of somehow coarse-graining from these smaller scales to the larger scales. But in some sense, the goal of trying to work on different scales is the hope that one day we can bridge from the smaller scale to the larger scale.
This is exactly what I'm interested in. Yeah. Very great to hear that. We have another question from Sylvia. She asks, I'd be interested to understand better how dimensionality was estimated in the first part of the talk. I understood that it is estimated by assigning all possible assignments to each object/data point. Why then does dimensionality decrease after shuffling labels of learned objects for good learners?
Right, that's a great question. That was something that we didn't expect to find. That was really something that popped out of our data analysis, and we were surprised, and then we had to try to understand it. So, one of the ways in which we can understand it is to think about how. Let's see if. Yeah. So I think the question was about why is it that we would expect dimension to go down. So firstly, we didn't know that this would happen, but you can think about it as that, because the shuffled data is sort of just looking at the neural data of the brain that say now doesn't have the information anymore. So it's just looking at how much resources is the brain using for that task, without being specifically to distinguish between different shapes, but just how much pure brain power is it using. And so because we saw that in the end that the fast learners actually had their null data, or this what we call an embedding data, because it should just be the data without any task information, that they actually had a smaller dimension, that it seemed that they were then using less resources, less just raw brain power, but then somehow they were coding, are able to distinguish between the different shapes when we put the shape information on better. So this is something that again, it's surprising for us as well. It wasn't, we didn't know or think that it would be there, but then we saw this, and so it's kind of cool that fast learners have both of these two things, both of high dimension according to the task, but yet smaller dimension of just their scrambled data compared to the slow learners. You're muted.
I'm sorry, there's a lag. After I click, I think it takes a while for it to get on. Okay. There's one question from Abhishek, and he's really excited, and we'll squeeze the time in for him. He asks, would mnemonic strategies affect these results? And in general, might these results also be indicative of the efficiency of underlying strategies adopted by fast learners?
Oh, that is such a good question. Sorry. Did I
and he has something else that he's saying after this, which is not a question, but it might be interesting to look at glymphatic flow in Alzheimer's disease to build a representation of the susceptibility of amyloid depositions to being flushed out through glymphatic clearance. Very cool talk. That's what he says.
Oh, wow. That is such a great comment. I had not thought about that. Wow. Thank you for your comments and questions. I think this thing about mnemonics is such a great question, and we didn't do work on that, and really this task isn't set up like that. But I would definitely bet that has to be the case, because people who have such good memory and such good memory tricks, they're really able to encode certain information in such a compact and efficient way that is so astounding compared to a regular person, let's say like me, who has to memorize even a phone number or something. That I'm sure that they must have some kind of memory device. And a lot of times you'll see that people who know these tricks, they're able to use the same kind of architecture and map on very quickly, even if you give them like a different thing to memorize, but they can use, they're able to just map that onto the strategies they already have and memorize that again very much quicker than somebody else who doesn't have these strategies. So in some sense, it is consistent with the fact that they probably have some way of already, some organization of the neural data that they can then use again and again as a strategy for memorizing things. And so we haven't done work on that, but I do think it's very related, and that would be a great direction to explore. And thank you for your suggestion about looking at proteins. Yeah, that is, I was thinking in some sense of these more signaling molecules, but of course, thinking about pathology could be a great idea as well. So thank you for that suggestion. It's great. And for your comments.
Okay. With this, we are done with our time here and also done with our questions. So perfect in time. Great. Thank you very much for the great talk. It was a very interesting perspective, which we as biologists sometimes tend to not go into too much. Just thank you very much again. And
well, thank
also
you for moderating. And I'm actually curious what you were saying, that you are interested also in this connection between the big and the small. And so I would love to hear also like from what aspect you're interested in that.
Yes, I am very happy to share with this, but after this talk. Great.
Okay, perfect. We'll talk after.
Yes. All right.
Thank you everybody who was listening and for all their great questions and comments.
I will end the broadcast then. Bye.